This is an overview of a recently completed survey project, ER19-1069, that studied the complete biodegradation of insensitive high explosive compounds. The project team was led by Jim Field at the University of Arizona. The co-PIs were Reyes Sierra Alvarez at the University of Arizona, Konstantinos Konstantinidis at Georgia Tech, Jim Spain at the University of West Florida, and Mark Krzymarczyk at Oklahoma State University. I am Osmar Menezes, a postdoctoral researcher at the University of Arizona, who, together with Erica Rios Valenciana and Camila Madeira, conducted research on this project. The Department of Defense is concerned about the environmental fate and impacts of newly developed insensitive munitions compounds, IMCs. These include NTO and DNAN. Previous investigations have established that both DNAN and NTO are subject to co-metabolic biotransformation in soil and waste streams, which generates an array of metabolites of poorly understood toxicity and increased solubility and mobility. The overarching objective of this project, however, was to develop an understanding of the mineralization processes that will enable the Department of Defense to deploy microbial strategies for the complete biodegradation of IMCs. These strategies could involve natural attenuation, biostimulation, or bioaugmentation at contaminated sites, or enable effective biological treatment of munitions wastewater. This project has led to an improved understanding of the mechanisms driving the microbial transformation of IMCs and reviewed the involvement of a combination of biotic and abiotic mechanisms in the degradation of these contaminants. We have demonstrated that some microbial systems are able to transform NTO and DNAN into benign mineralized products such as carbon dioxide, nitrogen, and ammonium, which eliminates concerns about the potential generation of toxic byproducts. Furthermore, we have obtained evidence for the anaerobic biotransformation of nitroguanidine. By providing shifting redox conditions in a sequential anaerobic aerobic treatment system, full biomineralization of NTO and of its reduced daughter product ATO to environmentally safe products was attained in both batch and continuous flow bioreactors. Moreover, this study provided multiple lines of evidence to support the occurrence of a novel mechanism of NTO biotransformation involving NTO respiration by Geobacter anodireducens. Our team has also discovered that quinone-respiring bacteria can be implicated in the degradation of a broad spectrum of nitroaromatic compounds beyond NTO. These microorganisms can couple the reduction of quinone moieties in natural organic matter to the abiotic reduction of nitroaromatic compounds by the hydroquinone moieties formed. The link between the microbial respiration of natural organic matter and the chemical reduction of nitroaromatics has important implications for the fate of IMCs in the environment and the development of environmental biotechnology processes. A sulfate-reducing enrichment culture containing Cupida sulfovibrio oxamicus was shown to transform nitroguanidine into nitrosoguanidine, which is further transformed abiotically to cyanamide and other unidentified products. This is the first description of a microbial community associated with nitroguanidine anaerobic biotransformation. The microbial culture utilized lactate as the electron donor, and both sulfate and nitroguanidine as the electron acceptors, and it could not utilize nitroguanidine as its sole nitrogen source. Experiments with synthetic munition manufacturing wastewater containing NTO and DNAN established the feasibility of constructing bacterial consortia with complementary degradation capabilities to achieve the complete biodegradation of these contaminants. We also developed a downflow soil perlite column with anaerobic microniches and aerobic zones, which allowed for the simultaneous biodegradation of NTO and its reduced daughter product, ATO. The results obtained in this project demonstrate that microbial processes provide promising perspectives for the treatment of munitions manufacturing effluents and for the in situ bioremediation of contaminated sites impacted by IMCs. This knowledge can be utilized to develop effective strategies to promote the microbial degradation of IMCs to environmentally safe end products. Our work also provides valuable insights about the potential contribution of microbially mediated transformations in the natural attenuation of IMCs. Additional work is needed to further validate the technical and economical feasibility of the proposed environmental biotechnologies and to establish design parameters for full-scale application. The future work should involve testing of real industrial effluents and contaminated media from the Department of Defense sites at the pilot scale. We appreciate your attention. Thank you.